Well, welcome everybody. I've got the very pleasurable task of emceeing this evening, so thank you all for being here. I'm Owen Ung and I'm the main chair of the Academy of Surgery, and Bernadette and I have sort of hosting you this evening, and we're all looking forward to your talk, Anne. Can I just begin by acknowledging the traditional owners of the land in which we're meeting today, the lands of the Turrbal and Yagara people, and on behalf of all of us here, pay our respects to ancestors and descendants who continue cultural and spiritual connections with country. We recognise their valuable contributions to Australian global society. Mark has been emceeing, Mark Smithers, who I'll acknowledge in the audience as a previous main chair. I took over the role from Mark, and Mark took over the role from Phil Walker. And Phil was the main chair when I came back to Brisbane in 2010. So I don't think I would be in this role if it wasn't for Phil's support at those early years. I'd just like to acknowledge all of you here, in particular Jeff McCall, our Executive Dean. Oh, actually he's not here right now, but I did see him earlier on. And of course, Bernadette will introduce Anne to you formally very shortly. But I'd just like to go through a bit of housekeeping before we get underway. Just a few small things. Now, for those of you unfamiliar with the main building, the toilets are located in the corridor either side of the lift and stairwells. And a reminder, if you haven't done so, please turn off your mobile phones to silent. And finally, if the unlikely event of an emergency, the fire exit is located to the right-hand side of the corridor outside the event space. So please follow the directive of our event staff in case there's an emergency. Now, I'd next like to introduce Doug Cavey to you. Now, Doug is a very close family friend and colleague of the Walker family. Had a conversation with Laurie, Phil's sister, who couldn't be here but sends her best wishes, who normally attends. And Doug won't remember, but when I was a medical student, he was my registrar at the Royal Brisbane, and I can remember harassing him until he let me do an appendectomy. And lo and behold, I had a career in surgery. So Doug, fond memories of my time as a surgical registrar, as a student, and you as a surgical registrar. But I will ask Doug to say a few words about Phil on behalf, as a friend and on behalf of the family. Thanks, Doug.
Thanks, Al. I'll be very brief. It's important to remember Phil. I see in the room distinguished people and colleagues and friends, and I also see a lot of students. And I don't think any of the students will know who Phil Walker was, so I need to tell you about him so that we remember him. Phil was a remarkable surgeon, academic surgeon, and outstanding clinician. He argued through his career that surgery was 20% science and 80% art. And the art of surgery is all the things we're going to talk about tonight. The ethical approach to patients, how you talk to them and listen to them, how you investigate new technologies and how you implement them. Phil was a vascular surgeon who did his fellowship in the Royal Australian College of Surgeons in General Surgery in 1987. Then the Australian Vascular Program, which was embryonic. Really, vascular surgery was a ill-defined specialty in the 1980s. And I did much the same thing, but I was in Brisbane. I didn't know Phil then. We both travelled to California, which was at the epicentre of this idea that you could treat a vascular disease, the largest arteries in your body and in your brain, by putting a pinhole in the groin. It's, what could possibly go wrong? It was a crazy idea, but it actually worked. And California was the beginning, really, of the major advances in technology. So Phil took a position in about 1990 at Stanford University Medical Centre as a fellow in vascular surgery for 2 years. And I worked at UCLA School of Medicine, Harbour UCLA Medical Centre in Los Angeles, 400 miles south, for nearly 4 years, 90 through to 93. So we didn't know each other, and we met in the ballroom of the Hilton Hotel in Chicago, I think. My wife, who's here, was there, and when I met Phil, he had a dinner suit and a feather boa on, and I thought, it's an interesting guy. And we became very good friends very quickly. Phil was an outstanding surgeon from the beginning, and he was involved in the design and the build and the insertion, the implementation of the first thoracic stent graft in the human in 1992, June 1992. So Phil and I both worked, not together, but in California together for some time, and then we both ended up back at the Royal Brisbane Hospital. We shared consulting rooms, the operating room, outpatients, a vascular lab, a non-invasive lab. Phil was the luminary, I followed along behind him. He was an extraordinarily good teacher, and I have all his PowerPoint presentations for students still in there, absolutely gold standard. And, you know, 25, 30 years old, they're still gold standard. A wonderful teacher. He became sick in his 40s with cancer at the peak of his academic and surgical career, and he remained quite unwell but working for 8 years. He died at age 57. He left money in his will for a large number of things through the College of Surgeons, Royal Brisbane Hospital Foundation, and for this lecture in perpetuity. The three ethical problems that Phil had with the way we should practice is how we treat patients on an interpersonal level, how we experiment and how we undertake research, which is the focus of tonight's presentation. And the third, but most important to him, was how you apply that to the community with equity, compassion and safety and consent. So now that you know who he was, I am very grateful for the time to be able to tell you, and he would have loved to have been here himself. Thank you very much.
Thanks so much for that, Doug. I'm now going to introduce Bernadette Richards to you, who's the Associate Professor of Professionalism and Ethics at the UQ Medical School, and she will introduce our esteemed speaker for this evening.
Thank you so much. Welcome everybody. Thank you so much for coming along this evening. It's my great honour to introduce our speaker tonight. And I was thinking about how I could introduce her. She's just achieved so much and has so much to share with us. And I think it's fabulous to see students here tonight as well. And one thing came to my mind today, and I thought, you know the saying, may you live in interesting times. Some people say that that's an insult, you know, a curse. I think Professor Kelso, in her time as CEO of the NHMRC, certainly lived in interesting times. She oversaw and guided revision of the grant programs as we know them and deeply improved them. Oversaw some significant changes regarding gender balance and other forms of equity in research. And from my experience, I had the great honour, really, of being involved in some of the work that is significant and that Anne will be sharing with us this evening, which was the review and implementation of mitochondrial donation. So this is why I'm saying interesting times, because we live in interesting times, and definitions of what makes a human, considerations of what we can and cannot do, and also more recently, which we just started to discuss, is really what is an embryo indeed, and the essence of that, and as I say, what we can and cannot do. So it's been a very interesting, challenging time, and some very, very important questions that will govern what it is to be human and the impact on Australian humanity going forward. And steered the ship and guided some very, very interesting and important conversations that will impact our future going forward. So we're a great honour to welcome you this evening, Anne. Thank you.
Well, thank you very much, Bernadette, and thank you, Owen. It's really fabulous to be here, and a great honour to give this Phil Walker Memorial Lecture. And I'm really delighted to have heard from Doug a bit more about Phil, and I really hope that what I'm going to talk about tonight is in the spirit of at least some of what he had tried to achieve and impart in his own work. I'm also delighted to be back at the University of Queensland Medical School. I spent a long time down the road at QIMR, 14 years, left some time ago. And you might not know that I actually held an honorary appointment in the Department of Surgery, despite being a cellular immunologist without a clinical degree. And you can imagine that caused some amusement for the medical members of my family, and they've yet to ask me to carve the Christmas turkey. So today I'll speak from the perspective of my many years as a biomedical researcher and also my more recent time with the National Health and Medical Research Council in Canberra, which was a very different aspect for my career. Now, I think as researchers, we tend to think what we're doing is important, that it's essential if we're going to solve the mysteries of human health and disease and find a solution to the challenges we face in our health. And for many of us, that's the reason we do research. And of course, it's the reason that governments fund us, that philanthropists fund us, that the general public supports medical research. And I think we're all beneficiaries of that very long-term investment in health and medical research. The average life expectancy from birth in Australia has increased from 71 years in 1970 through to 83 years today. So that's 3 months a year, every year for 50 years. That's an extraordinary increase in our longevity, despite the fact that all those major issues like quality of clean water were achieved a very much long time ago. And all of that is the outcome of research, which I think can be easy to forget. But health and medical research throws up issues of ethics and values all the time. And I think it's easy to gloss over those when we're so excited about the possibilities of medical research and innovation. Now, as I've stepped away from the lab bench, which I did quite some time ago, I've naturally thought about, needed to think more about the research system as a whole, my own very small place within it, and then also in my time at the National Health and Medical Research Council, NHMRC, to think more about what constitutes responsible and ethical conduct of research. And then I was involved, as Bernadette has said, in discussion of new technologies, particularly reproductive technologies, where I think ethics and values are just central to the decisions we make as a society. So I'd like to use the privilege of this lecture to talk about these experiences and, in a way, look at two aspects. I can call them two sides of a coin, they are sort of. The first is the ethics and values we bring to the conduct of our own research, and that will only be relevant to a few people in the audience, but I hope it's useful. And then the second is the ethics and values that we bring as a society to the translation of the outcomes of research into practice at the frontiers of medicine. Now, I'm not an ethicist, that's clear. I won't bring the kind of depth of thought and analysis that an ethicist would bring to these issues, but I will speak as somebody who's had, many decades living and working within the Australian research system. And I want to start with the left-hand side there, the hands-on aspect of doing our own research, how we design, conduct and communicate research. And here, I think the central question is, can the price of our research be justified by its potential benefits? Now, by price, I mean all of those things we put in, the public funds, the use of human subjects, of laboratory animals, and various precious materials, infrastructure, energy, not just the fossil fueled variety, of which a lot goes into research, but also researchers' time and energy, societal costs, the potential that we cause some harm to communities through the research that we do, or that we raise hopes inappropriately, or that we impose in some way, exploiting the goodwill of the community through our research. And then opportunity cost, anything you invest in this research is not available to be used somewhere else. And by benefits then I mean all of those good things on the right-hand side of the slide, which are the reason that governments and the public fund us. Now, this question of whether the price can be justified might not be something, and really is not something we can answer until the research has been done and it's been put in the context of other research. But I think we need, as researchers, to ask it from the beginning and all the way through, so that we are doing our best to tip that balance in favour of benefits over costs. Now, there are all sorts of codes and guidelines to help in a basic way with the way we think about ethics and values in research. NHMRC produces a lot of them, and NHMRC-funded researchers and institutions must abide by the Australian Code for the Responsible Conduct of Research on the left. If you're doing human research, you must abide by the National Statement, and then if you're doing animal research, you must abide by the Code for the Use of Animals in Research. And this is all well and good, it's essential. But if we are to do research that is ethically justified, there are issues I think we need to think about that go beyond what the considerations or remit are of an ethics committee. These aren't things that we can outsource to a committee. As we go through the development of a research project and its execution, I think we need to think about a number of things from this perspective. So first of all, choosing the research problem. Have we even defined the problem? Because it's very hard to solve it if you haven't defined it, and projects can very easily drift in this way. Does the problem need to be solved? Is there actually a gap in our medical knowledge? Is there an unmet medical need? And are we the people, and do we have the resources that are needed to solve it? When we're designing the project, will our design actually test the hypothesis? Are we choosing the right methods? Are we blinding so that we avoid bias? And of course, there are ethical issues if we're using human subjects, animals, and limited materials. Then when it comes to doing the research, do we actually know what we're doing? Now, there may be students here who've done some research in the lab, and when you first start, you absolutely do not know what you're doing. And so there's a learning process inevitably. But as more senior researchers, we need to make sure that our students and our research staff do actually know what they're doing. And then are we following protocols, checking reproducibility, are we recording data appropriately? And then when it comes to analysing the data, obviously we need to be using the right tools, and we need to be open-minded in interpreting the results. And then finally, a really important aspect is how we communicate the outcomes and whether we're describing the research fully and accurately. Are we publishing both positive and negative results? The tendency of the system is to focus on positive results where there's been a positive effect, but negative results are just as important, and it's critical not to suppress them. And are we citing other people's contribution to our field fairly? Now, research that is poorly designed, conducted, or communicated is wasted research. The price is not justified, and the research is not ethical. And actually, writing grant applications is good for us because it actually forces us to think about these questions in advance, even though we don't necessarily enjoy the process. Now, the use of animals and human subjects in our research is an area where many of us will at some point be considering ethical issues and submitting to an ethics committee. And most of my research was done with inbred strains of mice, which are beautiful animals with these different coat colours, which is actually why they were bred in the first place, but turned out to be really useful for immunological reasons. Now, that meant I needed approval from an ethics committee, and our funding didn't flow until we had that approval, and we were asked all the appropriate questions about whether we needed to use animals and how many we'd use and what techniques we'd use. All of that is really critical. It's exactly as it should be. But that doesn't answer the bigger question of whether the price of the research in the use of animals was justified by the outcomes, because the ethics committee can't answer that. And actually, I don't know the answer for my own decades of research. I believe that our work contributed to the body of knowledge. We contributed to the debates of the time. We published papers, we went to conferences and talked there. We were active in the research committee, community, the students, research assistants, fellows, were all trained and they moved on to jobs elsewhere and contributed in their fields. But that's not the same as knowing that that was worth the cost. But if I step back from there and look at research in immunology as a whole, then I have absolute confidence in saying that work with laboratory mice has transformed how we understand the immune system, and it has given us all sorts of opportunities to improve human health. And through the development of vaccines, through new drugs, particularly monoclonal antibodies, through the cancer cellular immunotherapies, in transplantation, in control of various immune and inflammatory conditions. And of course, if you discover something really groundbreaking, then you can probably say that the price was justified with the potential benefits as well. But for the rest of research, which is most of it, we know that it's part of the process of advancing knowledge, and you could say that it is intrinsically and unavoidably wasteful by its very nature, but it is an essential part of the process. What I'm actually more concerned about today is avoidable waste, and that is an issue of ethics. Now, back in 2009, a really important paper was published by Ian Chalmers and Paul Glasziou, and they reviewed waste from the literature in research, and there's a table in their paper which has some really pretty bad statistics in that are quite upsetting to read, I think. But for example, that more than 50% of the clinical trials that they were looking at didn't refer to systematic reviews to know whether the trial actually needed to be done. More than 50% didn't take steps to reduce bias, like blinding, and more than 50% were never published in full. Disappointing results were underreported, and so you're potentially suppressing important data in that case. Now, researchers may recognise some of these deficiencies in their own research. I think they're common. It doesn't mean that this sort of waste is, that it was all wasted, because much of this research will have been useful, but we can do better. And there is the question then, is it better today? And I think these discussions from people like Paul and others have been really important in encouraging pre-registration of clinical trials, and also efforts by some funders and the big journals to really improve the rigour of the data that are provided when people submit papers for publication. So I think that is helping. Now, the other upsetting thing that's happened over the last few years, another important wake-up call in medical research, has been the so-called replication crisis that some people may have heard of. Now, this was brought to our attention about 12 years ago by Glenn Begley and Lee Ellis, when Glenn and his colleagues who were working at a biotech company, Amgen, in the US, and were looking to develop new cancer therapeutics, and they found that they could confirm the published scientific findings in only 6 out of 53 really important cancer research studies. And from their point of view, it really turned out that being published in a top journal doesn't mean the research is necessarily of high quality and reproducibility. Now, they saw a range of issues that really come down to poor scientific practice, and they also talk about the pressure to publish, and so I want to come back to that in a minute. Now, I think that the work of people like Glenn and many others in other fields as well, such as psychology, has been really important in raising awareness of this issue. And again, just as with the waste work, I think it's been contributing to greater rigour in research and greater rigour in publishing standards and journal requirements. So, you know, there is a way to go, but these are really important things to be aware of. Because poor quality research not only wastes the resources used for that research, it also wastes the investment of others like Amgen in this case, trying to build on it, for example, to design a new cancer therapeutic. And I think this is an issue of ethics, not just economics. Now, these, I think, quite upsetting reports really point to flaws, not in the scientific process, but in our research system. But it certainly isn't all bad, and just remember, you know, how successfully research has improved the quality and longevity of our lives. This is not. We've had such extraordinary advances in disease prevention, diagnosis, drug treatments, surgical techniques and more. And I stand before you today as a beneficiary, particularly with my brand new titanium knees, so I am actually standing, so I'm very aware of the benefits of this sort of advance. But I want to come back to this question of the communication of research, and this is the slide I showed before. The potential gap between the research that we've done and what we say about it in papers and talks. Real research data, and as anybody who's done research will know, aren't always pretty and they're not always exciting. And there's a conflict between that reality and the pressures of a research career that really come to the fore when we're writing a paper. The decisions about which data to show, how we display it, the papers we cite from others, and the story we construct around the research that we've done. Now, we will naturally want to show our best results, although I do remember a student who liked to show his worst results in lab meetings, which always seemed quite odd, but most of us want to show our best results, and we'll want to explain away the experiments that didn't work the way we wanted them to, and we'll want to cite the papers from others that support our hypothesis rather than undermine it. But it's so critical, it's ethical, to present our research accurately and transparently, be clear about what are valid conclusions, and recognise other people's contributions fairly. These are issues where I think ethics and integrity and research quality are completely aligned. Now, cases of actual fraud, deliberate fabrication of research happen from time to time, and they harm us all. Not only is fraud likely to end the career of the perpetrator, it may lead to patient harm. It wastes resources in every sense. It may have damaged the careers of others in that research team and the reputation of the institution. And surgeons may be aware of this really dreadful case of a surgeon in Sweden who brought great shame on the institutions, including the famous Karolinska, where he worked. And so, this is not what you want to have happen. It undermines public trust. There's nothing good about a story like this, and it's really hard to understand why somebody would take the risks. But fortunately, these sorts of cases of outright fraud and fabrication appear to be rare overall, and they do appear to be rare in Australia. From what I've seen, the major case that some will be aware of at QIMR Berghofer in the last few years was an extreme outlier for Australia. And it was handled in an exemplary manner by the Institute Director, Professor Fabienne Mackay. Damage was surely done, but important lessons were learned and policies and practices were changed. Now, this diagram from NHMRC I think is quite useful because it makes the point that breaches of the Code for the Responsible Conduct of Research happen on a spectrum. And when we talk about these terrible cases of fraud, they're absolutely at this extreme right-hand end. We don't know how frequent they are, but they're rare as far as we can tell in the system overall. What I'm more interested in today is this grey zone, which I think we all have the potential to fall into, the sort of poor practices, the traps that we can fall into despite, I assume, being fundamentally honest in our research and wanting to contribute to push the field ahead. So let me say a bit more about the pressures in the system that might lead people out of the green and into this slightly hazy area of less serious breaches. The world of publication has changed enormously in my lifetime. And so here's a paper from 1975, which I have to admit was my honours year. And I was not precocious. So this paper came from the lab that I ultimately did my first postdoc in, and it's a typical immunological paper of the time. It's got what today looks like quite primitive data, very simple data, a handful of figures in the paper. There are no supplementary data because all the papers were just published in a hard copy journal. Online didn't exist. There are just a handful of authors of the paper. They all come from one institution, and it was all funded by one postdoc fellowship and one grant to the lab. Now that is completely different from the world today. So here's a almost randomly chosen paper out of the journal Nature in July this year. And you can already look at it with some horror and wonder if you're actually going to read this thing. But first of all, complex data. 5 figures in the main article that are all great big figures like that one. 9 extended data figures, 8 supplementary data sets online, lots of specialist techniques that needed special people to do them. Not surprising, therefore, there are 44 authors, 24 institutions in 6 countries, 20 funders. I saw after I'd chosen it, spent 3 years between being submitted for publication and being published. So they were asked to do a whole lot more work. No wonder there are 4 first place authors. I think that really shows the pressures that there are in the system, that you even need to worry about having acknowledgement of 4 authors. It's completely different from the sort of pressure that I experienced in the early years of my career. And I think we need to recognise how these sorts of pressures on people are drivers which can lead to poor research practice. They don't necessarily, of course, but we need to be aware of them. And that then undermines the ethics and integrity in research. We must work to counteract them. So training, supervision, team culture are all important. But even when these are good, we see this inexorable pressure to get novel and exciting results. And if you're an early career researcher, to have first author papers, hence the 4 first authors on the previous paper. And if you're senior, then to have last author papers, to publish in high impact journals, because it affects your chance of a grant, of a job, of a promotion, of maybe a prize. So I think we have many wrong incentives in our system today, and it's important we work out how to counteract them and change the system back to one that focuses more on the quality rather than these measures that could drive poor practice. So back to this question, can the price of our research be justified by its potential benefits? To summarise what I've said so far, what I think is that we need to understand our obligation to conduct and communicate our research to the highest standards of ethics and integrity. We need to do that to reduce waste, to improve reproducibility, and ultimately then to have a bigger impact through our research, which is the most important thing and what we are all, I think, wanting to achieve. We need, in our own teams, to be alert to the pressures that drive poor research practices and be aware of the incentives that really go the wrong way. And we need personally to model and recognise and reward best practice. And that's not only in our own teams and institutions, but it's also when we're doing peer review of other people's work. Now, breath. I want to flip the coins now and go on to this other side of the question, which is the ethics and values we bring as a society to the translation of research at the frontiers of medicine. And this is where I think medical advances can conflict with society's ethics and values. For me, some of the best examples arise in reproductive technology, and I want to talk about one of the cases that arose in the time I was at NHMRC. But before I do that, I want to just go back in history a little bit, and some people will remember this, for others it was before you were born. But to look at the extraordinary advances in reproductive technology over the last 50 years. The first IVF pregnancy in 1973, so just over 50 years ago. The first IVF birth, and Australia was a leader with the UK in this. The first donor egg baby in 82, the first frozen embryo pregnancy in 83, the first IVF surrogacy pregnancy where both the egg and the sperm were donor. And then the first cloned mammal from an adult cell in 1996. That, of course, was Dolly the sheep, looking very healthy here on the left and with her beautiful baby daughter Bonnie, born through natural conception and processes, on the right. That was a landmark. And these advances have continued with developments in stem cell technologies. The first isolation of embryonic stem cells from early stage human embryos in 1998, the development of induced pluripotent stem cells, which then bypassed a lot of controversy about using embryonic stem cells, and then the development more recently of mammalian embryoids, which are early stages of embryonic development that can be achieved in vitro, so bypassing the need for fertilisation of eggs by sperm altogether. Now, the major landmarks, like the introduction of IVF and Dolly the sheep and embryonic stem cells, were very controversial at the time. And I always think of this Lunic cartoon, which was in The Age in the early 1980s. And what we see here is an accident where there's been a collision of a truck carrying human ova and a truck carrying human sperm, and there is consternation on the faces all around. The clergymen are there wondering what to do. The bearded philosophers from Monash University have driven up in their van. Now, this cartoon probably offended some people at the time, and I hope it doesn't now, but it really captured the moral dilemma of the time. In the case of IVF, though, what was once ethically confronting for many people in Australian society is now completely normal. And so today we have about 100 accredited IVF clinics in Australia. We have more than had in 2021, which is the last official data that we have available. There were more than 111,000 cycles, and there were more than 20,000 babies born through IVF in Australia and New Zealand. So that's 1 in 18 babies in Australia born through IVF. And worldwide, it's estimated that there have now been 10 million IVF babies born since 1978, and about 1 million a year now. Now, in Australia and many other societies, any ethical concerns about IVF have been swamped by the success of the technology in producing healthy babies and the number of families for whom those babies have brought joy. So, developments in reproductive technology led to some very interesting legislation that's controlled the use of human embryos in Australia ever since. They're the Prohibition of Human Cloning for Reproduction Act and the Research Involving Human Embryos Act. Now, the critical barriers that are created by these two pieces of legislation are, first of all, that they prohibit the in vitro development of human embryos beyond 14 days, and that's really important for embryoids. Prohibit the human cloning for reproductive purposes, so there'll be no Dolly the human. Prohibiting creation of human embryos that contain genetic material from more than 2 people, and prohibiting heritable alterations to the genome, prohibiting the ability to transmit those alterations through to future generations. And then research involving human embryos or excess IVF embryos or embryos created by processes other than fertilisation, again, embryoids. These can only be conducted in Australia under a licence from the Embryo Research Licensing Committee, of which Bernadette was part, and is under the aegis of NHMRC. Now, many people see, well, perhaps not many, but certainly some people see this legislation as an impediment to medical progress. And it does prevent some types of research. But I think it showed real foresight in recognising the implications of future technologies. The legislation forces the Parliament to think carefully before allowing any sort of human genome modification that could be passed down to future generations. And I think that pause to think first is a good thing. Now, the case in point has arisen with mitochondrial donation, which is intended to prevent babies being born with severe mitochondrial disease. And I'll just run through this. I hope that most people understand about this topic. Mitochondria being organelles within every cell that play a key role in energy production and are therefore critical for healthy life. They're controlled by both nuclear and mitochondrial genes. Mitochondria contain DNA, and so whereas the nucleus of the cell contains the chromosomes that come from both parents and encode about 20,000 or a bit more genes, the mitochondrial DNA is only 37 genes, although there may be many mitochondria in a cell, so many copies, and they're inherited only from the mother because the egg provides the cytoplasm for the embryo. And when there's mitochondrial disease, it's due to there being mutations in the nuclear or mitochondrial DNA. There's low incidence, but they can cause very serious disability and be fatal at any time from early infancy to later in life, depending on the severity. So, mitochondrial donation is an IVF technology that can prevent this happening. and the intent is to prevent the transmission of mutant mitochondrial DNA from the mother into the embryo by using mitochondria from a donor woman. So there are several techniques for this. So what you can see at the top is the male and female, the mother and father, produce they have the egg and the sperm, but then there's the source of donor mitochondria coming from the green female donor there. And there are several ways you can do this, and on the left side you can see that you have both the eggs of the donor and the mother are fertilised, and then the nucleus is removed from the donor egg, and the nucleus from the mother's egg is transferred into that modified donor egg. So now you've got the nucleus from the fertilised egg is going into a cell where there are healthy mitochondria without a nucleus. So on the right-hand side, it's a very similar process, except that the nuclear transfer happens before fertilisation, and after the nuclear transfer into the donor egg, then there's fertilisation. So here we have potentially devastating set of diseases that are inherited through the maternal line, and we have techniques, as shown on this slide, that offer families with mitochondrial disease the hope of having babies with nuclear genomes from both the parents, but without the mutated mitochondrial DNA. So shouldn't we introduce this technology? I mean, isn't that just a no-brainer? Well, for many families, not all, but for many families with mitochondrial disease running through their maternal line, this is a no-brainer. But mitochondrial donation was prohibited by the legislation that I described a few minutes ago. Not permitted in Australia under the Prohibition of Human Cloning Act because it creates human embryos with genetic material from more than two people, because there are mitochondrial DNA coming from the donor egg. And there are also heritable alterations to the genome, because these changes will be passed on to the daughters of the daughters. It may also raise ethical objections where it destroys fertilised eggs, which is what is the case on the left-hand side of the figure. Now, there's only one other country which has legalised this process for mitochondrial disease, and that's the UK since 2015, only for severe cases, highly regulated, and very little data available. So what we know is that there have been fewer than 5 births by May 2023, and we don't know anything about the status of those babies and their health. And a lot of effort is being made there to preserve the privacy of the patients. But it does mean we are working in a vacuum to try to understand the impact of this technology. Now, the story started for us when a Senate committee recommended that NHMRC investigate and advise on a number of questions to do with this technology. And then the former Minister for Health, Greg Hunt, had a family in his electorate with a little girl, Maeve Hood, who has severe mitochondrial disease. So Minister Hunt was very keen that there should be introduction of this technology to enable families like Maeve's not to have children with severe mitochondrial disease. Now, this struck me, so he asked us at NHMRC to undertake a review of the scientific, ethical and social issues raised by mitochondrial donation. And I thought this was a really important thing for NHMRC to do. I thought it could be quite contentious, thinking back to those earlier controversies about IVF and stem cells and even Dolly the sheep, and that we would be crossing another frontier with this technology. I also found it ethically challenging myself, not on religious grounds, but because the technology was not properly tested and because it involves heritable changes to the genome. So that question of whether potential benefits outweighed potential costs was not resolved in my mind. So as we planned and undertook this review for the government, what I wanted to achieve was a neutral assessment of the issues, that we would do our best to explain the health problem, the technology, lay out the issues as fully and dispassionately as possible, and avoid in any way advocating one way or the other about the introduction of this technology. So here's what we did. We formed an expert working committee of terrific people and brilliantly chaired by Bernadette Richards. The committee produced the issues paper on the right, which you can find on NHMRC's website, and it's worth a read. We did a whole lot of public engagement, which I won't go into except one thing, and finally we did 2 reports on the outcomes of the consultation. And again, I think they really, both the outcome report and the expert working committee statement are really excellent documents that I commend to you if you want to know more about these issues. But I particularly wanted to mention the citizens panel we had. A group of, I think it was 18 or 19 people from all sorts of backgrounds who had no prior knowledge of mitochondrial donation, who came together over 2 weekends to learn about and to discuss and consider the issues around this technology. And they produced a statement in their own words, which again is really worth reading. So I think that with all of the information we got from the citizens panel and from the written submissions and from the advice of the expert committee, we got a pretty good idea of the issues. Now, there were a lot of issues, and I've just summarised some of them here. But on the pro side, there's the avoidance of suffering of a child and of their family born with this disease, potential to have children with nuclear DNA from both parents, which is extremely important to many families, potential to eliminate mitochondrial disease from future generations. But then on the negative side, there are unknown risks, and as I've said, we don't have data, don't have proper research data on the safety and effectiveness of this technology for humans, or the long-term risks. Take a long time to get the data on long-term risks for child and future generations. There are technical issues about the procedure that are not yet fully understood, would remain unknown. The child would have genetic material from 3 people, and this was something that was particularly interesting for the citizens panel, of what the rights and well-being would be of the child and of the donor, and what the impact would be on family relationships in this situation. And then potential use for unacceptable reasons, not to do with severe disease, the concept of designer babies, and the potential inequities of access to the technology. Now, there are more cons on that list than there are pros, but this is not a numbers game. There isn't necessarily a right answer here, at least until we know the outcome of introducing the technology. So what happened then? Well, the government created a bill which they gave the term Mave's Law, after Maeve Hood, in Minister Hunt's electorate. And this amends the legislation only for mitochondrial donation. So they've taken a cautious approach here. And it's now no longer an offence with this bill to create a human embryo for reproduction with genetic material from more than 2 people and heritable changes. And the bill is really important and cautious in this very important way that it's a 2-stage process. And in the first instance, all that has been legislated is research, training, and a clinical trial done at one centre of excellence, with the trial expected to take maybe 10 or 12 years. And then it will only be on reviewing the data from that trial that a decision will be taken whether to legislate now to allow this to be introduced to clinical practice. And then the licences are provided and all the monitoring happens through the Embryo Research Licensing Committee. So when the bill was considered, I was really impressed with the way the Parliament engaged. And there were private meetings with deep discussion, and then the debate in the Parliament was, I think, really quite considered, whether people were for or against. There were considered discussion, and members and senators took their responsibilities seriously. So you can see here what happened. They had a conscience vote, the first since the same-sex marriage bill, which was 4 years before. It was passed by the House of Reps, 92 to 29, and then by the Senate just before the government went to the election, 37 votes to 17. And so that new bill came into force in October 2022. Embryo Research Licensing Committee has been doing work. A grant was awarded to a consortium to undertake the trial, and hopefully enrolments for that trial will be possible before too long. But as far as I know, there's nothing publicly available about that. Now, I imagine if we'd had this debate even 10 years earlier, certainly 20 years earlier, it would have been more contentious. And I think the enormous uptake of IVF is part of the reason for that. Our society has already accepted a degree of intervention in human reproduction that we hadn't in earlier times. Our society also places a very high value on an individual's wish to have their own children, genetically related children. And the human stories from families with mitochondrial disease were really compelling in the consultation. I think we may not be so good yet at talking about medical risk, and particularly with the big advances in medical care, perhaps we have forgotten that not everything can be cured. And so it's hard to engage with the issue of what are the risks that come with technology like this. And I'm also very interested in the ready acceptance of genome modification in mitochondrial donation. So although individual genes aren't being modified the way they would be with gene editing, the genetic makeup of the embryo is modified, and those changes will be transmitted through the female line to future generations. So I think that by introducing mitochondrial donation here and in the UK, we have taken a significant step, and it does take us closer to accepting modification of the nuclear genome. In my mind, at least, not everyone agrees, but in my mind, the ethical considerations are rather similar. So why have I told this story? Well, I think that what we learn from this mitochondrial donation debate is advances in health and medical research can present ethical issues that leap ahead of community readiness to embrace them. I think for those of us who are in health and medical research or medical practice, we have an essential role in explaining this work and doing it in a way that is without bias, so that society can, as far as we can interact with it, can make good decisions. I learnt that the general community and parliamentarians are more than capable of understanding and thinking deeply about complex issues like this and reaching sensible conclusions, whether you agree with them or not. They are sensible, cautious conclusions about the cost-benefit balance. And I do think the introduction of mitochondrial donation will be relevant to the future debates on nuclear genome modification. So I've talked about two aspects of some very big problems. I'll just summarise by saying that I think when we go back to this fundamental question of the justification of the benefits by the price, if that's around the right way, ethics and values must underpin the conduct of health and medical research and its translation into practice. We can only measure those benefits in retrospect when the research has been done, but I think we have to work from the very beginning to ensure that the costs and the risks are as low as possible in how we do our research and in how we introduce new technologies to society and into practice. And we, whatever our role, we have expertise that means we should help society to come along on that journey by explaining current research and its consequences without bias, so that our Parliament and the people in general can decide whether to accept its price and realise its promise. So I want to thank the many people at NHMRC and on our committees, people like Bernadette, and the general community who engaged with us throughout that whole process. And finally, thank you for your attention in a long talk. Thanks.
Yet, and I think there's just a few minutes if anyone has any burning questions for Anne. Anything from the audience? There's a bit of time. I think you, Doug?
Wrong end of the day.
Yeah.
Fascinating, and the one word I haven't heard is lawyer. I haven't heard the word lawyer, which is refreshing, I have to say. What is the interaction with, I'm not talking about the legislators, but the legal community in this sort of complicated research?
That's a really interesting question, and Bernadette might be able to answer it better than I can, because we have people with legal experience from an ethics point of view on relevant committees, but that's not the same as engaging with the legal profession. And I don't really have an answer. We didn't engage in a specific way with the legal profession on these issues, and I certainly know that there are many who will be very interested in the path that was taken. It's a great question. Bernadette might like to add to it.
It was chaired by a lawyer.
Yeah, but an ethics lawyer. Yeah.
An ethical lawyer.
Yeah, ethical lawyer for sure. Yeah.
Thank you so much for a fascinating presentation. I am a lawyer and a qualitative researcher, and I loved your reflections on ethical research. And I was wondering if you had any in terms of what ethical research might look like from a qualitative perspective, over and above some of those factors you identified. Things like bias that you might not be able to eradicate in that kind of research, but are clearly really important with things like citizens' juries that you spoke of as well.
Yeah, I think there are areas of research which don't easily fit that idea of a blinded clinical trial. And certainly a lot of the research I did, which was not the type you're talking about, but was nevertheless in many cases very difficult to blind, and particularly if you're doing it on your own. So I think not all of those issues apply in quite the same way. But I think thinking hard about how to eliminate bias in the way you design experiments or studies and the way you minimise bias in the interpretation of data and the collection of data, that point is still valid. And usually I think the answer is partly in how you share that with other people so that you're not entirely on your own sitting in a dark room choosing which data to show. There are such traps that we can fall into if we don't make sure that we have systems in place to protect us from our own biases.
Once again, thank you for your presentation. I'm not too sure how this question will come out, so forgive me if it's a bit confusing, but as you presented yourself, there's been great progress in reproductive technologies, and lots of questions have been raised about what it means to be human and what we're allowed to do ourselves. And being researchers, I'm sure you and your many colleagues are quite familiar with the field, but I think from a layperson perspective, it can be quite overwhelming, and often there's a sense that a lot of work is being done outpacing perhaps the ethical considerations, maybe not so much by the research committees, but by the people of the Australian public and their own perceptions. And so I guess that's about the concept of readiness there. Is there anything that people within the medical profession should do to allay some of that concern, and is that something that medical professionals should be doing in the first place? Is that the job for someone else, perhaps?
Well, perhaps we tend to think it's a job for somebody else, but maybe it starts at the kitchen table, and that we all in our own lives have opportunities to talk about these complex issues where we might have expertise and we're surrounded by friends and family who do not. And when these big issues come up, I think it's really good to get in there and talk about them. And I think just as our experience, for example, with the Citizens Panel, you learn so much from hearing how somebody who's not caught up in the world of research or does not have a deep understanding of medicine, to see how they see what's important. So as individuals, we may not all be able to get out into the world and talk about these things, but we can at a kitchen table level or with our friends over coffee or whatever. I think we can. In the case of the consultation for mitochondrial donation, lots of people engaged with that, but they were still only the people who were interested. And I know that for people in my own family, they didn't know about it, they didn't hear about it. So it's really difficult that we have something which I think is quite important that ended up going through the Parliament, and that the vast majority of people would not have been exposed to those issues and would not have been able to say to their Member of Parliament, for example, I think this is great or I think this shouldn't happen. So that the messages that come to our MPs are primarily from the advocates for or against, and not from the general community. So that's a difficulty and a frustration, I think, that I'm not quite sure how to overcome, except that we all do it at our own micro level. It's a good question, thank you.
Thank you.
Thank you, Anne, for a wonderfully stimulating conversation, even at this time of the night. Your talk did touch on the ethical and even the moral obligations that we have when we do these kinds of research. And I know the studies in the UK and in the Australia that we're collecting data. I wonder whether you can comment on the ethical and moral obligation to share that information across countries.
Yeah, yes, we could have a good conversation about that. And within NHMRC and the committee, we did engage as best we could with the regulator in the UK to find out what we could, and they shared a certain amount, but it was very clear that their primary concern was protecting the privacy of the individuals. And even when it was really because of a freedom of information request that some data were released that a first baby had been born, that was, I think, about a year after the baby had been born. So it might be a bit different here. I don't know.
Thank you very much. I wonder if I could just ask you about how we engage with journals and journal editors, because fundamentally most research is designed to be presented, and it's summarised in a very succinct format of a small number of words with a small number of tables, but also has to show some sort of positive outcome or it won't be published. What sort of discussions, I mean, that introduces a whole bunch of biases, and so what sort of discussions should we have with journals and editors to liberalise research communication?
Yeah. I think that this is one of the fundamental issues. Unfortunately, some of the journals are really embracing it, so the nature journals and some of the others, BMJ, I don't know about New England Journal or all of the big ones, but some of the journals are really concerned about it. They've been the journals that have been publishing papers like the Begley and Ellis one and aware of the issues. I think in many ways it's got better because of all that dreadful supplementary data. So now you do have the opportunity to show a lot of data. And I know that some of the journals are being much more demanding in the details of materials and methods that they require. So, is that just a checkbox exercise, or are they really taking it seriously? I think we're seeing a slow revolution, and it is getting better. Unfortunately, this bias towards positive results for publication is really hard to get around, and so there are journals which don't have that bias, but of course they don't have the high impact factors that everybody's chasing, and this is the problem. We're using as, if you like, surrogates for excellence the number of papers that people publish and whether they publish in high impact journals. And that ends up driving these difficult behaviours, I think. So some of the journals are working on it. It's hard for any individual researcher to change what a journal does, but I think that some of the big journals are really coming to terms with their responsibility here. In the meantime, just to extend a whole other area that I haven't talked about, but there are all of the fake journals. And so you've got a handful of journals, high quality journals, really trying to make a good system. And then you've got this great pile of journals which are completely fraudulent and which are easy to publish in, and they'll pay you to publish in them, and they'll give you your positive reviews just like that overnight. They are totally fraudulent, and they're really contaminating the research record. So there's a whole other world there that we have to tackle and be aware of as well to improve the quality of the research record.
I recall reading Biomed Central, they had once had an idea where we accept your protocol and then we'll guarantee in that to publish the outcome, positive or negative. Yeah,
so pre-registration of research is becoming much more the thing, certainly for clinical trials, and that's been the case for a long time, but there's much more of a drive now to have other sorts of protocols of research studies pre-registered so that you say in advance what you're setting out to do, and then if you've got some journals, as you say, the BMC journals will promise to publish the results, then I think this is really positive. But it is a big cultural shift, isn't it, when we've all been focused on getting that paper in Nature.
Thank you, and but the last task is really just to thank you for a terrific lecture that no doubt has from the question and discussion has been very stimulating for the audience. Some budding young researchers in the audience and some very experienced researchers in the audience. I guess thinking about the big questions in research, but also some deep thinking about the conduct of that research. So you really stimulated some thought. Thanks, Anne. And finally, thank you, Natasha, for your organisation for this this evening. Everybody's enjoyed greatly, so I wish you all safe travels. Thanks again.